My name is Guo-Ping Shi from the cardiovascular divisions from Brigham Women's Hospital here, local, in Boston. Attended this 21st International International Conference of Cardiology meeting in Boston. I speak about the interactions between asthma and abdominal aortic aneurysms. Abdominal aortic aneurysm is a very risky disease in the aorta. Affects men above or peripherally in men above 50 years old, or those aged peoples. And the problem of this disease is number one, there's no medications available, and surgical repair is the only way to deal with it. Secondly, once there's such a disease happens, and very easy to lead into sudden death because of the ruptures. Therefore, it is very important to have biomarkers to predict the risk. And the topic that I'm going to talk, what I talked about is the interactions between this disease with other diseases, for example, diabetes and autoimmune disease, psoriasis, for example, and we study the role of asthma affects the event of this disease. And we start off with humans, patient samples. We got about about 20,000 patients that has aneurysm patients, and we found that patients among those populations with aneurysms, patients with asthma tend to have higher risk of ruptures, sudden death, significantly higher risk. And in a separate population of patients, we found also about 16,000 patients from Denmark. We found that patients with asthma are prone to develop abdominal aortic aneurysms, which is we call it AAA in short. Therefore, in humans, in both populations, we proved or we established a role of asthma affects this aortic disease. So to test the mechanisms and to prove the concept, we used the mouse models to develop asthma and aneurysm at the same time, or one disease after the others. No matter which combinations you do, we found that mice with asthma, they tend to have higher risk of developing abdominal aortic aneurysms. So exactly we can recapitulate that what's happening in humans, and we thought that's the observation from humans are real. We can replicate it in mouse models. The mechanism behind this phenotype is because of asthma patients or animals, they have high level of IgE and high level of mast cells. That's another topic that we do in our lab. And those IgE, for example, is the key player in asthma, and also in the AAAs. We found that IgE can activate the mast cells, not only mast cells but also macrophages and the vascular cells, and cause them death or release those cytokines. Those are molecular mechanisms, and then link those two diseases together.